Thank you for joining us, Dr. Lemire. Dr. Lemire is a medical doctor and a clinical fellow for Care for Rare in Canada. And, you know, her main specialty is a KAT6B disorder. And recently she had a publication about KAT6B, a very nice summary about the KAT6B disorders that she published. So welcome and thank you. Thank you for joining us tonight.
Perfect. So just let me know if you don't hear me well or you don't see my slides anymore. But like Natasha said, so I'm just going to give you a little bit about my background. So I'm just going to admit people. Good. Okay. Just make sure I don't, everybody can join. So I did my medical training and my residency training in medical genetics in Montreal, Canada at the Université de Montréal. During my training, that's where I met Dr. Philippe Campeau, who was one of my supervisors. And Dr. Campeau worked a lot on KAT6B disorders. He was one of the first scientists to actually found the gene that caused the condition that are associated with KAT6B mutation. So that's by working with him that I became interested in KAT6B and the genetic condition that it's causing. So that's when I started to do this work on KAT6B disorder. Once I completed my my residency training in Montreal in 2019, I did after that 1 year of research fellowship within the Care for Rare Research Canada Consortium, which is a program that investigates rare genetic diseases and try to find new genes and diagnose individuals and patients and families who have undiagnosed rare genetic diseases. And after I did that, that actually since last month, now I am actually in Cambridge, Massachusetts, because I'm working at the Broad Institute in Cambridge. I'm doing a research fellowship also on rare genetic diseases. So I'm working on specifically on a project that's called the Rare Genome Project, if ever any of you have heard of it. So I'm a genomic analyst for families with undiagnosed rare genetic diseases at the moment. I met Natasha actually by email last year when she emailed me about the KAT6B Foundation Day that was supposed to occur in March. And I was happy to join. And now this is the same presentation that I would like to give you today instead of the one we were supposed to have in March. Okay, excellent. So here my presentation was more for towards families. So there's a lot of kind of explanation. It's more about talking about the clinical features of this condition, and for sure at the end we can discuss if you have questions or anything I could maybe answer if you have additional questions. So just to start, so KAT6B, when we look at OMIM, which is kind of an online catalog of gene disease associations, we see that KAT6B gene is associated with actually two conditions. One is called genital patellar syndrome, and I'm just gonna. Okay, so genital patellar syndrome, and the other one is called SBYSS syndrome, and that's these two conditions that we're going to talk about today. So the first one, Odo syndrome, was first described as a genetic condition that was characterized by intellectual disability in association with congenital heart disease and also dysmorphism. And by dysmorphism, what we mean is a variant in morphology, so different trait that can be kind of grouped together and seen more often in a specific genetic condition. And after this first Odo syndrome was described, after that, Young and Simpson in this article in 1987, so that's more than 30 years ago, they reported a kind of a different phenotype that were referred as the C. Barber-Biesecker-Young-Simpson syndrome. So a very long name that now most people call SBBYSS. And this particular condition was characterized by what we see here on the picture of this individual, so blepharophimosis, so having smaller opening of the palpebral fissures. So the eyes are actually of normal size. It's really the opening of the palpebral fissures that are smaller. Also having dacryostenosis, so meaning that the lacrimal duct, so where the eye drop are leaving the eyes, they are actually closed, so the communication is not correct, is not made correctly, and also ptosis, so meaning that the eyelids are also drooping over the eye. The other thing that was mentioned in this article in 1987 is that the individual also had a mask-like facial appearance, which means like a face with a little bit less expression, and also long thumb in grade 2. So that was the first time that the SBBYSS syndrome was reported. And after that, more in 2000, then the genital patellar syndrome was also reported, and it was characterized by hypoplastic or absent patella. So this is when the kneecap is smaller or just not present, and also with contractures of the hips and the knees. So when the joints are actually limited in their movements, as well as agenesis of the corpus callosum, which is a structure at the midline of the brain that was just either smaller or not present, as well as microcephaly, and also genital anomaly. And it's only later in 2011 and 2012 that exome sequencing, so sequencing of all the genes of an individual, it identified that mutation in KAT6B, that gene, was actually causing both GPS, so genital patellar syndrome, and also SBBYSS. And my past supervisor, Philippe, was actually involved in finding this gene. There's actually two groups here that found it at the same time and published kind of in the same, almost the same journal at the same time. And this one was found in 2011. Just to give you maybe a little bit more for families, that I wanted to just maybe explain a little bit more. If you know these things already and you can stop me, but I just wanted to give you kind of a little refresh about a little bit of what this gene is doing in cells and in the body. It's a gene that is involved in what we call epigenetics, and that's the way that the DNA helix that we see here, that's the way that the DNA is actually condensed and packaged so that it can be into a much more condensed structure than if it was a loose DNA strand. And this is important because this is linked to the function of this KAT6B gene. We talked about histone acetylation. So the histone are these small circles that you see here. It's a protein on which the DNA strand is going to wrap around. And this is one of the ways that the DNA gets condensed. And usually what we see is that there are various states in which the DNA can be packaged. And when it is loose, like here, what we see is that the histones are more relaxed, more loose. And there are these groups here that are the acetyl groups that are added to the histone, and that's what's making the histones more loose. Whereas when the groups, the histones here are more condensed, they have more of these groups here that are called methyl groups that are added. And this is important because the job of the function of KAT6B is to add these acetyl group here. And when the histones and the DNA is more loose and relaxed, that's when the cell and the what we call the DNA machinery has access to these to the DNA strand and is able to read the DNA, is able to read the DNA and create and produce what we call an RNA, and the RNA is then translated into a protein, and the protein is actually really what is making the instruction in the body, in the cell. So here it's another way to show it here, so KAT6B, what that means is that the K here is for lysine, and then acetyltransferase is the name of the gene. And what that means is that it's transferring an acetyl group on a lysine, and a lysine is just like one of the segments where on the histone where something can be added. So when you see here, mostly on histone 3 here, you see that there is this little kind of blue flag here that can be added, and that's exactly what this gene is doing. So this KAT6B is creating a more loose histone, and so the DNA is more relaxed, and it's what it does is that it's influencing the way genes are read in our body, and the way that so then more genes are transcribed. And this particular KAT6B works in a complex with other proteins, including KAT6A. So that's why these two disorders are kind of also sometimes discussed together, even though they have differences. So during my residency training, when I told you that I started to work on this condition, so one of the things that I did with Philippe Campeau and Dr. Lee, who works in Texas, is that we wrote a gene review. So this is, maybe you've heard of it before, but it is mostly an online resources on genetic diseases for clinicians, but can also be of use for families, but it's more of medical terms that are included in this review. And the other thing that we also did is that we produced what we call a case series. So we published the clinical information of 32 individuals who have a KAT6B disorder. So I did that with Philippe Campeau and also Lixin, which is a medical student in Montreal. And we had also many collaborators from many countries who shared the information about the individuals that they follow who have a KAT6B disorder. And what this allowed us to do, so this to better understand what we call the phenotypic spectrum. So that means what are the actual clinical features that are seen in people with KAT6B mutation, and also better understand what are the mutations that they have exactly in the KAT6B gene. And this was published in May of 2020. So this is what I'm going to talk about mostly today, kind of giving you a summary about the article and the findings that we had. So as I told you at the beginning, GPS and SBB-YSS have been historically described as two different conditions. But then we realized that with time, they have several overlapping features. So both GPS and SBB-YSS, they both include global developmental delay and intellectual disability. They include hypotonia, also genital anomalies, and patellar hypoplasia or agenesis, heart defect, dental anomalies, hearing loss, and thyroid anomalies. So these things can be seen in both. However, there are still ways to differentiate both conditions, even though they are caused by the same gene. So usually, when someone has two of these major features here and a mutation in KAT6B, this person is said to have GPS. And when someone has two of these features here in this list, this person is said also to have SBB-YSS. As you can see, anomaly of the patella is seen in both. So this can be another thing that's overlapping, can be seen in both conditions. And also, there are many articles that have been published in the past 8 years about that gene and individuals who have mutation in that gene. And what has been emerging is that there's also an intermediate phenotype. And that's when individuals have 2 features of each usually. So it's really kind of they have really features of both conditions, and it's also a mutation in KAT6B. So we cannot tell if it's SBB-YSS or GPS. So then they are called in the literature as having an intermediate phenotype, and that's also how we described it. So we have made a huge table actually in the articles that I tried to subdivide here so that maybe it's easier to see. So let me know if you don't see anything here. But I just wanted to go through a little bit about it's kind of a summary of the clinical features that were found in individuals with GPS, SBB-YSS, and their intermediate phenotype. So in our cohort, but also from all the individuals in the literature, which is at approximately 90 people that were described in past publications. So one thing that we wanted to point out here is that so in our cohort, there were 3 kids who had, when they were while they were a fetus, so while their mom was pregnant with that individuals, when they measured the nuchal translucency, which is something that is measured at around 12 weeks of gestational age, this nuchal translucency was increased. And also these 3 individuals had a cystic hygroma. So that's when the nuchal translucency is increased, but also we can see kind of pockets of fluids on ultrasound. So that's something that had not been described before. And in here we have it in 3 individuals out of 32. So that's still a significant number. So that's just something that that should be added in what we call the differential diagnosis. So meaning when during pregnancy, when this sign is identified, just to know that KAT6B disorder could be a cause for that, for clinicians who follow pregnancy. And another thing that was found during pregnancy is that so in our cohort, 2 pregnancies, and in the literature, 5 other pregnancies were they were complicated by polyhydramnios, which means that there's too much amniotic fluid around the baby without any other reason. So these were the 2 anomalies that we saw more in terms of prenatal findings. Otherwise, here what we see is that so almost all individuals in the cohort had global developmental delay or intellectual disability. There was also a language disorder in at least 60% of the individual. And of these individuals who had, I'm just sorry, I just saw something on the chat. Okay, thank you. I just read that. Okay, thank you. What I was saying, so all of the individuals who had language disorder, 25% had hearing impairment as well. So for sure, these 2 things can be related as well. And here, what I wanted to show is that almost all individuals with a GPS had agenesis or hypoplasia of the corpus callosum. But like we said, this is an important feature in that condition. Seizures were really rarely observed, so only 11% in the literature, and very few individuals in our cohort also had seizures. So this is something that happened less frequently here. If we continue, the other thing that was found is that in total, actually 20% of individuals had conductive or sensory neural hearing loss. So hearing impairment that was found, and it's not necessarily at birth, it's something that can also develop after. The other thing that we have here in the table is the blepharophimosis. So that's I'm going to show you pictures, more pictures of soon as well, but that's the smaller palpable fissure. So what we can see here is that it's something that is really seen in SBB-YSS and the intermediate phenotypes, but not seen in individuals with GPS. Also, the lacrimal duct anomaly that is here. So it's something that is a bit more rare. That is seen mostly in SBB-YSS. The other thing that we found actually when we analyzed all of our 32 individuals is that so about 25% had a cleft lip or palate, and the other thing also to follow was the dental anomalies. So some kids had dental delayed eruption of teeth, or also absent or hypoplastic teeth, so having smaller teeth than expected. So that's the dental anomaly here. That was more frequent in the SBP-YSS also. And this is the last part of the table. So lots of information here. I'm sorry, there's a lot of text. But one other thing that needs to be followed for individuals who have KAT6B mutation is the thyroid. So we see here that there was hypothyroidism, so the thyroid that functioned less. Here that was found in 38% of individuals with SBP-YSS in the literature, and also in a few individuals in our cohort. The other thing also is that needs to be evaluated is genital anomaly that were seen in both, actually all 3 subtypes. If we see here, also for sure a bit more in GPS, because as I said, it was one of the criteria to also clinically classify these individuals with having GPS. And this can include very different type of genital anomalies. So in males, this can include cryptorchidism, retractile testes, scrotal hypoplasia, micropenis, and hypospadias. So very, very diverse. The other thing also that should be evaluated is the kidneys by a renal ultrasound. And renal anomalies were more commonly seen in GPS individuals. And the 2 most frequent things were multicystic kidneys, so having kidneys with cysts that can impact the way the kidneys are functioning, and also hydronephrosis. So that's when there's a dilation of the ureters of the kidney. And if this is severe, this can also impair the way the kidneys are functioning. Anal anomalies also have been seen in about 6 individuals. So this is a rarer anomaly, but this can be having do you have a question? Should I continue, or is someone that?
Yeah, I think so. I think so.
Yeah. Okay.
Yeah.
So so basically the anal anomalies I said was rare, but what can happen is that to have anal atresia, so really having the anus that is not present at birth, or that is actually stenotic, so being smaller, so it's harder for the baby to have normal feces. Another thing that can happen with the digestive system is what we call intestinal malrotation. So that can lead to having the bowel that kind of twists on itself. That has been reported only in individuals. So this is not a frequent features, but that's something to kind of that we need to have more information about if this is really something that we should be looking for in our patient or in individuals who have a KAT6B mutation. And the last thing I wanted to show is that here there's contractures and spine anomalies. So especially for GPS, this is something that should be looked at. Actually, no, the real last thing is that half of individuals in all subgroups can have a congenital heart defect. So, and most oftentimes, these are not serious malformation. It can be just a ventricular septal defect, which is like a hole in between the two chambers of the heart. But because of that, we recommend that all individuals with a KAT6B mutation have at least once echocardiogram, so an ultrasound to look at the heart. This is so I wanted to show you clinical pictures of about 20 individuals that were in our cohort, that were from various places, various countries around the world. So here what you can see is that in SBB-YSS and also in some kids with intermediate phenotype, so you can see the blepharophimosis, so having like smaller palpebral fissure. And also what you can see is that some individuals have what we call the mask-like facies, so having kind of a face that has a bit less expression, and this can evolve over time as well. So that's something that can be kind of present when the individual that's something that can happen. I just want to admit person. Well, that's something that can happen, that can be present in babies, but then when they grow up, so for example, this little girl here, when she's older, this is not present anymore. And the other thing that we see here is, so especially for SBB-YSS, one of the features that can be seen is the long thumb and the long great toe. That has no impact on the way the hand or the foot is working. It's really just something that has been noticed that is more frequent in individuals with SBB-YSS. Here, in individuals with GPS, we see that, so this is the absence of the kneecap. So that's why we see a dimple here. We can also see contractures in the club foot here, or contractures of the knee as well. And that's what we see as well here. We see absent hypoplastic kneecap. And here I just wanted to maybe just, because I didn't know if you knew the difference. Let me just, okay. So I'm going to talk about the genetic variant in the KAT6B gene. So, and I just wanted to maybe give you a kind of a review about the different type of genetic variant that we can have in genetics. When you have a DNA sequence and you change a letter, because that's mutations are like typos, so you can have what we call a missense variant. That means that it's changed the meaning of the DNA sequence, and every DNA sequence has a meaning of, so they are read 3 by 3. So that's what we call the codon here. And that's translated into an amino acid, and amino acids are the building blocks to create a protein that will ultimately do the function in the body. So when you have a missense, is that when the letter has been changed, it's changing the amino acid. So that's, you start from one amino acid here, so and it's becoming another one, and that can change the meaning of the instruction that the gene is asking for. But for KAT6B, most of the mutations are actually nonsense. And by nonsense, what we mean is that you have a DNA sequence, and by changing a letter, so here we see that we change, there's an A here that's not present here. So you replace the C with the A. And what that causes is that when the body or the cell reads that gene, it just stops reading afterwards. So that just stops the protein there, and all the rest of the sequence, so the rest of the amino acids that were supposed to be there, are not included in the protein. So that's what we call a stop. And the other terms that we see often with KAT6B is frame shift. So frame shift is actually what it causes is that it's also leading to a stop, but not right away. So the nonsense here, we have a stop, like the letter is changed, and we have a stop right away. So the cell stops reading the gene, sorry. But here, when you have a frame shift, it's that you changed, you have a new letter, for example, that's changing the way that's changing the codons, and then the meaning of the sequence is different, and ultimately what it leads to is that it leads to a stop. So then the protein has incorrect amino acids that have been added, and then ultimately it's also shorter because the cell stopped reading the gene at some point. So I just wanted to explain that to you because this is a diagram that's showing all the, so here it's all the reported KAT6B mutation in the medical literature. And here below is the one we had in our articles, the one that we had for our 32 individuals that we reported in this article. And what you see here is that most of these variants are actually stop, so nonsense, or frame shift. So when you see the little star here, that means a stop. So that means that after this position, then there's no more protein. So that's a nonsense mutation. And when you see FS here, it means it's a frame shift that's causing a stop 11 amino acid after that. So this is the two main type of protein here. You can also have larger deletion that include like a bigger part of the gene, and that would also cause the same problem, meaning that the gene instruction would be missing. If we look a little bit closer here, so what you see is that the colors associated with the actual mutation are associated with the disorder. So orange is for SBB-YSS and blue is for GPS. So what you can see on this figure is that the mutation that are leading to GPS, they occur most oftentimes here at the beginning of exon 18. And I should probably tell you that the small boxes that you see here are exons. So exon 3, exon 12, exon 18. The largest exon is this one, exon 18. And the small line that you see here are introns. And this is the way genes are constructed. And once the cell reads the instruction, what they do is they only keep the exon, and by a mechanism called splicing, the introns are removed, and only the DNA letters that are in the exon are actually included in the final protein. So the exon contain really what we call the coding information. So if I come back to what I was saying, so that all the mutations that are causing GPS are actually located mostly here at the beginning of exon 18. And the variants that are associated with SBB-YSS, they occur throughout the gene. And they occur more often in exon 16 and 17. So you see here the orange associated with SBB-YSS. But then you can also have some that are more at the beginning of the gene. And what we can see is that, in general, just to finish, so SBB-YSS, you can also have individuals that have SBB-YSS, but that are actually in exon 18, but more at the end of the exon 18, like you can see here with the orange lines. And the last thing is the green is the intermediate phenotype, and that's when I was telling you is that individuals have kind of features of really both conditions at the same time. And in general, what we see is that in individuals who have SBB-YSS, more at the beginning of the gene, so like these mutations here that are orange, they tend to be a bit more mildly affected. Whereas individuals who have SBB-YSS caused by a mutation in the last part of exon 19 tend to be a bit more severely affected. And by severely affected, I mean oftentimes having more malformations. So having malformation of the hands or the fingers, and having more genital urinary malformation as well. There are no variants in the literature or in our cohort that have been associated with both GPS and SBB-YSS. So there's no variant, no mutation that cause both, but there are mutations that are being reported that cause either GPS with the intermediate phenotype or SBB-YSS and the intermediate phenotype. So that's what you see here when you have a circle that has two colors. So it's either here intermediate or SBB-YSS, or here you have blue and green. So this is GPS and intermediate. So most individuals who have a KAT6B disorders, they have a de novo mutation. And by de novo, we mean that the mutation starts with the individual. So it's not something that has been inherited from parents. But in the last few years, there have been 2 families that have been reported that it was a familial case that was milder, actually. So here we have a large family with many individuals that were having that were affected by SBB-YSS, and they were mildly affected. And same here by another group. It's Kim and collaborators in 2017. And also in our cohort, so we also had a mother and a daughter. So individual 30 in our cohort was the mother of individual 10. The mother was actually diagnosed when we diagnosed her kid, and then looked in the variant for her. So this individual had a global delay and also the smaller palpebral fissures, and also the long toe and great thumb. But the mom only had learning disabilities and no diagnostic of intellectual disability. So she was for sure a more like a milder case than what has been reported in the literature. But what we're learning is that for sure GPS, SBB-YSS, and the intermediate phenotype, it's all part of a broad clinical spectrum. And there are for sure more individuals that are affected but just are very mild, and that's why they're not reported in medical literature. But now we're learning more about these individuals. So in conclusion, the management of individuals who are affected by a KAT6B disorder. So these are the recommendations that we came up with. So at initial diagnosis, so when the individual is diagnosed with a KAT6B disorder, we recommend that there should be like a physical exam for evaluation for genital and anal anomalies. So that can be done by a pediatrician or any provider that can do a physical exam. Contractures should also be evaluated clinically, and then X-rays or consult in orthopedics could be done also if needed. We recommend that at least 1 echocardiogram and 1 renal ultrasound be done, because there can be subtle anomalies that could have been missed earlier, or that could not necessarily show any symptoms when the baby is still young. Also ophthalmological evaluation for the malformation that can be seen with SBB-YSS, but also there had been a few individuals who had also retinal differences. We recommend also a thorough neurological evaluation with a cerebral MRI, and also only a clinical surveillance for seizures, just because it's still a rare feature. So we don't think it's important to absolutely have an EEG. So the electric recording that neurologists do, it's only if there is a suspicion of abnormal movement. And lastly, we think that their hearing should be periodically evaluated. So if there's any doubt about hearing, that then an audiogram should be done. And then the thyroid also could be checked probably annually to make sure that there's no hypothyroidism. So a thyroid that functions a bit less. And on that, I'd be happy to talk with you and see if you have any questions.